And we have Dr. Zhiyue Wang giving a presentation today on nutrient removal and resource recovery from municipal wastewater. So just want to turn it over to Tom for a little bit. Our director, Tom Gianbaluca, give some opening remarks.
We're really thrilled to get started this semester with actually two new faculty members at Water Resource Research Center. One is Aurora Kagawa-Viviani, who's I think on the Zoom, and Dr. Wang. Zhiyue is our newest faculty. Technically, he's just joining us, and so this is a great opportunity for us to get to know him and his research. So I'm not going to take up any more time. Just turn it over to.
So with that, I'll introduce our speaker. Dr. Wang is a new faculty member at WRRC and the UH Department of Civil and Environmental Engineering. He obtained his PhD in Environmental Engineering and Science from Stanford University and was associated with the Reinventing the Urban Water Infrastructure Engineering Research Center. His research interest focuses on reducing greenhouse gas emissions of municipal wastewater infrastructures and improving the sustainability of nutrient management with novel resource and energy recovery biotechnologies. So with that, I'll let you take over.
All right, thanks for the introduction, Tom and Gary, and nice to see you all in person. My name is Zhiyue Wang. Nice to meet you all. I think thank you so much for inviting me here for the seminar. I think this is a great opportunity to introduce myself as well as my past research experience, and then maybe we can see, I'm seeking potential collaboration opportunities if anybody is interested in my work. So the title of my presentation today is Nutrient Removal and Resource Recovery from Municipal Wastewater. So let's get started. So first, why do we need to worry about nutrients? So nitrogen and phosphorus are actually two of the most seriously crossed planetary boundaries defined as the safe operating space for sustainable development. And it is a global issue. So on the right-hand side, you can see areas with highlighted in red and yellow are the areas with increasing risks of nitrogen and phosphorus flows into the ecosystem as well as ocean. So poor nutrient management can actually pose risks to ecosystems as well, experiencing like eutrophication, acidification, algal blooms, and along with other social and economic losses. So I've only been here for a month, but I noticed there's also a problem here in Hawaii. So we have a lot of cesspools around, sipping actually 1 million gallons per day of untreated sewage into the ground and also into the ocean. Along with it are not only nutrients, but also pathogens and other micropollutants. So all this pose great threats to the recreational use of the water, as well as the safety of the groundwater that we're using it as a portable source. In order to solve this problem, let's take a closer look at what's inside municipal wastewater. So this is my very rough log scale of the substrate concentration inside wastewater from left to right. So on the right-hand side, we're seeing very high concentration of organics, salts, usually over hundreds of milligrams per liter. And then there's our nutrients, nitrogen and phosphorus, which is usually 20 to 50 milligrams per liter. And then on the left-hand side, which is very low concentration, usually ppm or ppb level, those are the metals or micropollutants. Even though they're at low concentration, they still pose very high risks to public health, as well as health of the ecosystem receiving untreated wastewater. Such substance includes pathogen and also other emerging contaminants like pharmaceuticals, chemicals, and even microplastics that's generated from our daily use that goes down the drain. So this municipal wastewater, the problem of this municipal wastewater is further complicated by energy crisis, water deficiency, and also climate change. So in order to approach this problem, we need to have an integrated mindset, turning wastewater actually into a source of clean water, energy, and nutrients. So again, on the left-hand side, that's the scale I have for the substrate concentration. It is also a problem of across scale. On the right-hand side, we're seeing that's the size of the treatment system. So we have like 1 to 10 people. That's a household size for on-site treatment systems, all the way to a centralized system with existing collection infrastructure for such as wastewater treatment plants that can go up to millions people population. So these different systems actually require different solutions and different biotechnologies to recover these resources from wastewater. For example, at smaller scale, we're looking at septic tanks, membrane bioreactors, whereas larger scales, we can do anaerobic digestion inside a wastewater treatment plant, and also reverse osmosis to recover clean water. So these are very different ways that we can tackle with this problem. And then since this is a very complicated, it's complicated in nature. So actually National Academy of Engineering identified 3 out of 14 grand engineering challenges that's related to municipal wastewater. That includes managing nitrogen cycle, providing access to clean water, and engineering tools for scientific discovery. So today in this seminar, I'm going to share some of my past research experience and my research work trying to address these challenges that's related to nitrogen, water, and engineering tools. That includes nitrous oxide recovery from wastewater, and a technology called microbial encapsulation, and lastly, some of the mathematical and molecular tools we developed along the way to study these engineered systems. So first, let's take a look at the nitrogen cycle. So current anthropogenic nitrogen cycle are very energy intensive and unsustainable. So if we look at where we get our source of nitrogen, most of them are from the atmospheric nitrogen gas. And we use the Haber-Bosch process that consumes about 19 kilowatt hours per kilogram nitrogen, fixing gaseous nitrogen from the atmosphere into ammonia and use it as a fertilizer. And then the fertilizer part of it gets lost during agricultural process, part of it goes down the food chain and pass on to us. And then we excrete it into wastewater, municipal wastewater. And when it goes down the drain into the wastewater treatment plant, the conventional wastewater treatment process actually consumes another 4 to 6 kilowatt hour per kilogram nitrogen and convert this ammonia, waste ammonia, back into nitrogen gas and release it back into the atmosphere with no value recovered. So if we take a look at what's happening inside a wastewater treatment plant, this is a simplified pathway for nitrogen removal. So we are starting with a most reduced form ammonia on the top left corner, all the way oxidizing to nitrate. So this process requires a lot of oxygen. So we are aerating it, mixing it, so it consumes a lot of energy. And once it's converted to nitrate, there's also a lot of organic carbon requirement to reduce it all the way to nitrogen gas. So otherwise, this organic carbon can be used to produce methane for energy recovery. So this pathway made us think, can we come up with a better design of the nitrogen cycle? Is there a way to bypass this slope of oxidation and downslope of denitrification? So we found out actually there's one intermediate inside along this denitrification path, that's nitrous oxide. So this is a better oxidant than oxygen. It decomposes to nitrogen and oxygen gas at the same time releasing energy. So can we shortcut this pathway and have nitrous oxide as the end product for our treatment, and then combine it with the biogas coming from anaerobic digestion of methane. So the co-combustion will actually produce more energy than combusting methane alone in air. So we went back to the drawing board and then calculated whether this makes sense, whether this is feasible. So this is what it looks like for a conventional nitrification, denitrification system addressing, including nitrogen and carbon flow. So if we're starting with like 1 mole of ammonia, and then we need to pump a lot of oxygen into it to oxidize it to nitrate, at the same time producing biosolids. And then we need to divert part of this organic carbon in the wastewater into reducing nitrate to nitrogen gas, at the same time producing a lot more biosolids during this process. And then the remaining carbon that we can use it for anaerobic digestion, and then converting it to methane for energy production. So in general, we are putting in a lot of oxygen into this process, producing a lot of biosolids, and then recovering some energy from methane if we have anaerobic digestion in place. So on the right-hand side, we're showing a redesigned nitrogen cycle that produces nitrous oxide as an end product for nitrogen removal. So again, we're starting with 1 mole of ammonium, and then we're putting less oxygen into our oxidation process because we're stopping at nitrite. And then we're diverting also less organic carbon into this process to produce nitrous oxide only. And both of these steps produce the less biosolids than the conventional treatment process. And then we have more carbon, organic carbon remaining in the stream that can be used to anaerobically digestion to convert it into methane and produce more energy. So this process will produce actually 60% more energy than the conventional one on the left, and also reduces the biosolids by 40%. So Dr. Skerson in Dr. Criddle's lab demonstrated this process first in 2014 in lab. So prove that this route is possible for a shortcut nitrogen removal pathway. And then they named it coupled Aerobic-anoxic nitrous decomposition. In short, CANDO, fancy name. And then they demonstrated it with a 2-reactor system. So on the in the first reactor, ammonia is converted into nitrite, which is a process that's been studied a lot. A nitritation process. And then the second reactor is the one that actually produces nitrous oxide. It's running in a sequencing batch mode. So it has 2 phases. In the first phase, anaerobic phase, where acetate is added, bacteria will take up acetate and store them inside their cells until the anoxic phase, when nitrite from the first reactor is transferred into the reactor to initiate anoxic phase, and then bacteria will use their stored internal carbon to reduce nitrite into nitrous oxide. What we found is that in this way, decoupling the addition of carbon and nitrogen, we're able to selectively produce nitrous oxide as the end product, and then we're harvesting it at the end of the cycle for energy recovery. So this is what we observed from a typical cycle of an operational cycle of the second reactor, the CANDO reactor, producing nitrous oxide. So as I described just now, there are 2 phases. In the first phase, anaerobic phase, we're observing acetate being taken up by the bacteria until it's depleted. And then there's a peak concentration of stored carbon inside the cell, that's the blue line. And then we're transferring nitrite from the first reactor, adding it in, and bacteria are reducing the nitrite to nitrous oxide while consuming their stored internal carbon to produce nitrous oxide. And nitrous oxide is harvested at the end of the cycle at this peak level. So we identified the key intermediate for this process as polyhydroxyalkanoids. So it's a polymer that has two most common monomers, including a C4, a 4-carbon monomer called hydroxybutyrate, in short HB, and a 3-hydroxyvalerate, that's 5-carbon monomer, in short 3HV. And then our hypothesis is that the bacteria will take up volatile fatty acid, in this case acetate, including acetate, propionate, butyrate, all that stuff. And then they can take it up and then store it inside their cell as granular polyhydroxyalkanoid, or PHA. And then during anoxic phase, they will, when there's only abundant nitrite but no extracellular carbon, they will use their internal storage as a carbon source, as well as reducing equivalent to reduce nitrite into nitrous oxide. And indeed, in the lab scale reactor, we found a lot of these organisms like Commomonas, Paracoccus, Diaphorobacter. All of these are denitrifying bacteria that has the capability of storing PHA inside their cell. So since this process looks very promising, we pushed the technology forward and did a pilot scale testing at a local wastewater treatment plant. So this is a 1,000-time amplification from bench scale to pilot scale. And this reactor on the right-hand side is 5,000 liters in one reactor size. And then it's set up actually below the anaerobic digester, receiving its centrate. That's very high concentration of ammonia inside. This is done in a Delta Diablo wastewater treatment plant in California. And then through this pilot testing, we're actually able to shorten the operational cycle from a 48-hour cycle to 24-hour cycle, and also increase the reaction time 5-fold with their, increase the nitrogen load, increasing the nitrogen load of a kilogram nitrogen per cubic meter per day. We're able to achieve about 0.2 kilogram nitrogen per cubic meter per day. So that is comparable to other existing nitrogen removal technology in the field. Something interesting that we observed when we increase the loading of nitrogen is that another monomer appeared inside the polyhydroxyalkanoids, the PHAs. That is the HV monomer, the C5 monomer we talked about. So in order to track its origin, we conducted a 13C isotope label study. We added 13C labeled acetate into the feeding it to the bacteria, and then did an anaerobic batch incubation. So what we found out, and then when we harvest the cell, extract the PHA, and conducted the nuclear magnetic resonance spectroscopy, we're seeing that the 13C labeled carbon actually were only incorporated into the HB monomer, the 3-hydroxybutyrate monomer, not the hydroxyvalerate monomer, since it's only showing up on the 13C NMR, but both of the monomers showed up on the hydrogen, the proton NMR. So that made us thinking there's some other source of carbon inside the bacteria that's providing this carbon chains to the polymer PHA. So our guess is that glycogen can be another source of internal storage compound that's participating in this nitrous oxide producing process. So we hypothesized that this denitrifying glycogen accumulating organism that acts very similarly to what we thought before contains both glycogen and a PHA. And then they also take up VF, the volatile fatty acid, during anaerobic phase, with that's assisted by glycolysis, that's the degradation of glycogen. And then they can use the PHA as the electron source and also carbon source to grow and produce and respire nitrite at the same time, producing nitrous oxide. And during this process, glycogen is actually replenished during the anoxic phase. So they're ready for the next cycle of volatile fatty acid addition. Again, indeed, in lab, we found out after sequencing, the reactors were dominated by a Diphthococcus-related bacteria that's known as glycogen accumulating organism. So again, chemical analysis also confirmed that glycogen participated in this process. So we're seeing, again, a typical operational cycle inside the nitrous oxide producing reactor in the can-do process with two phases, anaerobic phase and anoxic phase, where acetate, when acetate is added, we can see a very rapid uptake of acetate that's in yellow. That's assisted by the consumption of glycogen, which is represented in the green line. That's the measurement of total carbohydrates. At the same time, we're seeing production of both PHB and PHV in the PHA that's extracted from the cells. Again, when we add nitrite to it, we're seeing the consumption of PHA during this process and the conversion from nitrite to nitrous oxide. At the same time, glycogen is replenished back to the level they're ready for the next level, the next cycle of operation. So we believe that when we reduce the cycling time in the pilot scale and also shortening the operational cycle, we'll select for faster growing or microorganisms that actually takes up acetate faster, that's assisted by the degradation of glycolysis. So besides glycogen accumulating organisms, there are also other microorganisms that can accumulate internal storage compounds, such as polyphosphate. So other research groups are currently actively seeking such possibility for poly-P accumulating organisms to participate in nitrous oxide production. So that will allow simultaneous removal of nitrogen and phosphorus, at the same time producing nitrous oxide as end product for energy recovery. There's also some engineering challenges left for such process, so that we can harvest nitrous oxide more efficiently since it's a dissolved gas. And then we need to optimize the reactor configuration, as well as explore the application of can-do process for lower concentration nitrogen feeds. For example, the mainstream wastewater, that's usually low concentration of ammonia. So the next biotechnology I'm going to talk about is microbial encapsulation. Let's shift gear a little bit. So this is some of my past research trying to address the second challenge, engineering challenge, that is to develop a new biotechnology for both water and wastewater treatment. Again, we're looking at the graph that this graph here that's showing this problem is across scale. At smaller scale, we're looking for technology that's low cost, self-sustaining, and very small footprint. And at larger scale, we have these huge infrastructures that are usually energy intensive, larger footprint, and then produce a lot of biosolids. So we need to come up with new biotechnologies addressing these issues. So one of the technology I've been working on is microbial encapsulation. So that has this biotechnology actually have some of the advantages across different scales that can address these issues. So microbial encapsulation is actually using a permeable matrix to fix bacteria in place. So on the left-hand side, we can see they will create a barrier between the encapsulant and the bulk liquid. And then there's mass transferring, allowing substrate and the electron acceptors to diffuse through the encapsulant and then allowing bacteria to grow inside. So when you have a high concentration of cells fixed in place, that will enable not only faster treatment rate, and it will also allow faster settling because they're encapsulated in these fast settling beads. And all of these will allow to have a more compact reactor design for a treatment system. And because of this mass transfer creating concentration gradients of substrate and electron acceptors, these will create microenvironments inside the encapsulant, allowing a diverse microbial community to survive. And then it also protects them from any inhibitors in the bulk solution, making such process more robust. And lastly, since we are able to select what kind of microorganisms we want to encapsulate, it can be tailored for different applications, which I'm going to talk about in the next slide. So this microbial encapsulation is a platform biotechnology that can be applied to various environmental applications. For example, resource recovery. If we encapsulate anaerobic bacteria, we can convert waste organics into methane and hydrogen for energy recovery. At the same time, we can also encapsulate fermenters to convert organics into glucose and ethanol for biorefinery purpose. And lastly, there is also possibility that we can use the chemical property of encapsulant itself to recover heavy metals from the waste. And as for nutrient recovery, we demonstrated that encapsulating a diverse community containing both ammonia oxidizing bacteria, denitrifiers, and also Anthropogenic bacteria inside a single encapsulant will allow a higher removal efficiency as well as a more compact single reactor design to achieve complete nitrogen removal. And for remediation purposes, we can do a chemical or biological, we can add chemical or biological degradation of the contaminants by dosing active compounds or active bacteria that can carry out the job while retaining them using encapsulation over time. So this technology has also been tested across scale. Since we're talking about the scale of the system, so Dr. Novak's group has been testing a pilot scale system in the brewery using encapsulated reactor to treat brewery wastewater in Minnesota. And this will be an example of a distributed system where they can convert the organics in the brewery wastewater into hydrogen or methane so that they can recover it for energy use. And then some other companies actually are already also in the market trying to develop commercial solutions using encapsulation technology. So for example, Microbial from California, this is a system treating drinking water, polishing its water quality using this kind of bubble tea kind of beads. And then there's also Biocastle from Israel. They're trying to install these cages that contain encapsulated bacteria and then dipping it into the existing wastewater treatment plants to enhance its treatment efficiency. And lastly, Hitachi has been working on this for decades. They are trying to develop a nitrogen removal system with encapsulated bacteria, removing ammonia from the wastewater, and then they can be used to upgrade existing infrastructure like a activated sludge process by dumping these beads into the existing aeration tank, then allowing a faster removal and better water quality from the treatment system. So lastly, I hope to share some of my research addressing the third engineering challenge, that is to engineer tools for scientific discovery. So with a rapid development of both molecular and molecular technology as well as computing power, I think we've been trying to use mathematical modeling to tackle a lot of engineering problems. And this becomes very handful in process optimization as well as understanding the mechanism of existing or a new biotechnology process. So at the same time, we will be needing new analytical tools to validate the model predictions, so that we can go through this iterative process by extracting important key parameters from reality and construct a conceptual model that allow us to measure parameters, and then through simulation, giving us predicted results based on our understanding, and then validated against reality. And then this iterative loop will allow us to have more accurate prediction and optimization of the model. So in the case of nitrous oxide production, based on our hypothesis of denitrifying glycogen accumulating organisms, we are able to establish a metabolic model that describe the interconversion of polyhydroxyalkanoates and glycogen. So also explaining why we expect the 3HV coming from glycolysis. And we incorporated that into a mathematical model that separately simulate a soluble substrate that's external, the extracellular soluble carbon, for example, acetate. And then they're metabolized differently with when you compare it to an internal storage compounds like glycogen and PHA. So with this simulation, we're trying to predict all the flows of carbon and nitrogen, the key parameters in this nitrous oxide producing process. So here are some of the results from the model prediction in dashed lines, and the dotted lines are the experimental measurements. So the model actually successfully predicted the changes of key concentrations of the key intermediates for both carbon and nitrogen, for example, acetate, PHA, and glycogen on the bottom figure, and then nitrite and nitrous oxide concentration on the top figure. And with this tool in hand, we are able to optimize the process based on model prediction. And then actually we doubled the nitrous oxide yield, at the same time shorten the reaction cycle from 24-hour to 12-hour. And then by harvesting nitrous oxide at the highest concentration inside the reactor. So in the case of encapsulation, we hope to better understand the microbial growth inside the system with modeling. So we incorporated mass transfer and also the property of encapsulant into the model, and trying to predict their consumption of substrate, in this case ammonia, production of nitrite, and then their encapsulated growth inside the system. So the model actually accurately predicted these rates and conversions, and this is again an overlay of experimental measurements and the model prediction. Also predicted by the model is that there's a minimum zone of dissolved oxygen as well as pH near the center of the encapsulant. So the bottom two figures are showing across time. There's the y-axis is the radius, so 0 means the core of the encapsulant that actually developed a lower zone of lower concentration of oxygen, and that could potentially inhibit the growth of ammonia oxidizing bacteria. So this information will help us optimize the reactor design and also guide our selection of encapsulant, and both of which will help us enhance the treatment efficiency. As for the validation of the model predictions, there are some of the existing analytical tools can provide some information, but not sufficient enough for us to validate the results. For example, microscopic tools that can be used across different scales that give us information about size of cell clusters using electronic microscopy, SEM or TEM, and then all the way up to optical microscope that's looking at the morphology or the surface of the encapsulant. And then this technology actually cannot distinguish live or dead cells, and also any distinguish between different microorganisms that's appeared. So we're only seeing cell clusters mostly inside the matrix. There are other molecular tools such as high throughput qPCR that can tell us quantitative information of different groups of microorganisms that's being encapsulated and how they change over time. However, there's the information of depth profile is lost during that process, because usually we're homogenizing the sample and then measuring the extracted DNA out of the sample. So we cannot tell any, we cannot obtain any information on the distribution of the cell inside the encapsulant. So we decided to develop a new tool for the validation of our model information. So we tried to develop this cross-section fish method. The top row is showing the process, how we prepare the sample. So for example, we're using alginate as the encapsulant. In this case, we have these beads that encapsulate ammonia oxidizing bacteria, and then we fix the cells using PFA, and then embed them into a sliceable matrix. That's GMA. That's a plastic-like material. And then after embedding them, we can cut them using a microtome into very thin pieces. And then these we can add fluorescence dyes that's targeting different groups of microorganisms using fish to look at the different groups of microorganisms and their spatial distribution on each slide under fluorescence microscope. So the bottom one is what we observed over time. It's a little hard to see, it's pretty bright here. But the red are the DAPI stain, like that stains all the bacteria as a counter stain. And the green ones are the ammonia oxidizing bacteria lit up by the fluorescence probe. So over time, we're seeing at the beginning, we started with very small colonies that's evenly distributed, because that's how we prepare the beads, by mixing them up. And then over time, we're seeing this ring formation near the surface. That's the colony growing near the surface of the bead. And then after day 20, the integrity of the alginate actually starts to break down. And then we see that on the breakup of the structure over time. And then there's more bacteria growing on the surface of this collapsed structure. So we actually pushed this tool further and trying to extract depth information from encapsulated growth. So this is how we can set up the experiment. So how we use the tool to dissect the entire beads every 100 micrometer. And then we're harvesting 3 consecutive slices and using it as a triplicate for each step. And for each of the slides, we can use different fluorescence probes to target for different groups of microorganisms. In this case, we studied a more complicated microbial community that contains not only ammonia oxidizing bacteria, but also nitrite oxidizing bacteria and anammox bacteria using different type of fluorescence probe. And this is what we're seeing typically under a microscope in a single slide. So for each of the slide, we have a square area of imaging located at the center and looking at different colonies that are growing inside the encapsulant. And then with that, we can obtain information such as count of the colony as well as the area of each colony, so the size of each colony. And then with if we take pictures along the axis, we'll have a stack of images that can measure the distribution, spatial distribution, and also size distribution of different groups of microorganisms inside a single bead. So this is still unpublished data. So we have, this is just showing the ammonia oxidizing bacteria colony count and size over the steps. So x-axis is depth, 0 means the surface of the encapsulant, and then about 1200 is the core of the encapsulant. So we're seeing there's actually preferential growth near the surface of the beads, starting from 200 micrometers. And the three colors are three different triplicates that's obtained. We're cutting three different beads from a single reactor. And then the count tells us information about how many colonies are there. And on the right-hand side is the size of each colony detected, so how large they grow. And we're observing that there's largest colonies can be found between 200 and 500 micrometers in depth. That means this region is actually the optimal space for ammonia oxidizing bacteria to grow when there's also other presence of other microorganisms. So this information allows not only to validate the model predictions, but also shows a potential competition between different microorganism groups, and also optimize the encapsulation, the design of encapsulant in future applications. So yeah, in summary, so I showed some of my past experience, my projects aiming to address these three grand challenges, and I hope to continue my research in this area. On the right-hand side is a figure I really liked that nicely summarized approaches that I like to use in engineered microbiomes. That is to build one, test it out, learn it, and then go back to the design board, optimize it, and start this iterative loop. So when approaching these problems, I'm very open to solving problems with multidisciplinary approach, including chemistry, microbiology, process engineering, and also mathematical modeling. So all these approaches greatly helped my research navigate its way to this iterative design process. So in my future research, I'm hoping to continue my work on nutrient removal and resource recovery while developing new technologies for clean water, and also developing new engineering tools along the way for scientific discovery. So lastly, I would like to acknowledge some of my advisors, my PhD advisor, Dr. Craig Rideau from Stanford, and my postdoc advisor, Dr. Paige Novak, and Dr. Satoshi Ishii from University of Minnesota. And these are some of the funding sources for the research projects I just described. And thank you for your attention. And here is my contact information. So please email me. All right, thank you.